Hello everyone. Now we're going to use some of those definitions that you've learnt in the last video, and we're going to look at how characteristics gets passed down from generation to generation. Now you need your notes book, and we're going to be looking at page 5 and going on all the way through to page 8. So page 5 introduces us to a gentleman called Gregor Mendel. Now, he was an Austrian monk who lived in the middle of the 1800s. And in those days, monks lived in monasteries and they grew their own plants and looked after their own animals. And Mendel was a keen gardener, and he was more interested in how plants grew, but he was also interested in how characteristics got passed down from generation to generation. And he noticed that when he was growing pea plants, that there were, most of the pea plants were tall, but there were some shorter or dwarf ones. He noticed that usually the tall plants had tall offspring. And the same with the small ones, they bred amongst themselves and produced small plants. Now, he did an experiment, and this really was the first experiment in genetics. And your textbook and your specification talk about a monohybrid cross. So a monohybrid cross in genetics just means two organisms reproducing. So like a mother and a father are crossed. That means they reproduce and they have offspring, which is you. All right. Monohybrid, what does that mean? Well, mono means one. Hybrid is really like a coming together of two forms. So if you think of hybrid cars, when we talk about hybrid cars, we mean cars that have an engine that works on electricity or on usually petrol. So obviously some more modern cars are all electric. Other older models are all petrol, but a hybrid one, the engine can run on either electric or petrol. Sometimes it's diesel. So in genetics, by a monohybrid cross, what we mean is that we're really just looking at one. The mono bit is one characteristic. Okay, so one characteristic, which in this case was the height of the pea plant. Now later on you might do crosses looking at eye colour, or maybe you might do crosses looking at whether people can roll their tongue or not, and look at examples of monohybrid crosses there. But as I say, this guy, Gregor Mendel, he was the first person to work out really the mathematics behind genetics, and thanks to him we can do a lot of calculations and do a lot of research into how things get passed down from generation to generation, and that kind of genetics is called Mendelian genetics. And most people find it really interesting. So getting back to our pea plant, we took some pure breeding tall plants. What do we mean by pure breeding tall plants? We mean tall plants that were allowed to breed with each other for a period of time, and they always produced tall plants. So we call them pure breeding tall plants. And then he took some small plants that when they reproduced amongst themselves, they always produced small plants. And so you need to write, it's really the first blank on page 5, pure breeding small plants. So these plants are referred to pure breeding small plants. So I'll give you a few seconds to get that written down. So what Mendel would have done whenever these plants were flowering, so this is just a diagram, it's not actually what a pea plant looks like, but say they've got a nice colourful flower. He would have taken something like a paintbrush and taken the pollen from one and put it into the flower, the female part, the stigma of the other, and vice versa. He would have done it the other way around as well. What did he find out? Well, what he found out was, these are our parent plants. We call that the parental generation. And these were the offspring. So when they were crossed, that's why we've got a little cross there. When they were crossed, he found out that all of the offspring were tall. Now this next generation is called the F1 generation. The F stands for the word filial, which comes from the Latin word which means son. So it's really the F1 generation. So what he did then was to take two of these plants and breed or cross them. Okay, so two of these plants were crossed. And he found that 3/4 of the offspring were tall and 1/4 was small. Now, if this was the F1 generation, we call this next generation the F2 generation. Now, when he did a little bit of thinking, this meant he basically decided that every pea plant must have two kind of, well, he called them determiners, two factors or two forms, which is carrying the information for height. And we can see then in the pea plants that you can have tall plants, you can have small plants. So he decided that each individual must have their chromosomes. You get one chromosome in each pair from each parent. So one from your mum, one for your dad. It's the same in pea plants. They get one of the chromosomes from one parent and one from the other. So that means that you've inherited one gene for height, or these pea plants inherited one gene from height from one parent and the other one from the other parent. Now, gene for height, Mendel said, came in two different forms, tall and small. So they're both coding, they've got the bases that code for the proteins that will make tall plants, and the other ones have got the code that will basically make the plant be smaller. Now, as we said, each parent passes on one chromosome, so each parent is going to pass on one of these forms. These forms of the gene are called alleles. So hopefully you remember that word, not only from the video, but also from year 10. So we've got a gene for height, and there are two different alleles. One is tall, and the other is small. Back to our experiment, these were the original ones. So tall and small. We're saying that the parents have got two alleles for height. And because these were purebred or pure breeding, we can assume that both of the alleles were tall. In this case, both the alleles here would be small. When reproduction happens, one of those goes to each offspring. So each of those is going to have a tall allele. And one of these small ones is going to go to each offspring. So if these were our parents, and this is our F1 generation, we can say the F1 generation, those plants must have had two alleles, but one of them was tall and one was small, but yet the plant grew to be tall. So that tells us that the tall allele is always expressed. It's like a set of plans, and that's the plans that are always put into action. And we say that this one is always expressed. So this must be the dominant. And the small one, Well, it's there, but there's no sign of it. It's basically ignored. The opposite to dominant in genetics is recessive. So in this case, the small one is the recessive allele. What about when these two were crossed? Well, the tall goes to half of the offspring, and the small goes to the other half of the offspring. So we've got a tall, small, small. And then if we look at these parents, tall is going to go there. And it's going to go there. And small is going to go to the other ones. Now this is very messy, but remember what we said? We said that the tall is the dominant allele. So that's tall, that's tall, that's tall. Small is only expressed if there's nothing else there. So this one here is small. That one there's tall, even though it's half of the alleles are tall, the other half are small. It's like a set of plans. If you have a set of plans to build, let me see, a shed. You've either got the set of plans to build a shed or you haven't. You might have a set of plans for something else, but if you want to build a shed, you need that set of plans. In this one here, we've got two sets of plans to build a shed. Well, we can build the shed, so we build the shed. Same here. Same here, we've got the set of plans for the shed, so we'll build it. So those three have got the plans as such to be tall, and so they are tall. This one here doesn't have the plans to be tall, so it ends up being small. Now, hopefully I haven't confused you more, but we talk about alleles. And so what we're saying is that the tall plant had two tall alleles, the small plant in the parents initially that was purebred had two small alleles. We know that the tall allele is the dominant one. And so what we do when we're doing monohybrid crosses is to pick one letter. And you can pick any letter you want, but it makes sense when we're talking about tall plants. We might as well pick the letter T. We could have picked any letter, doesn't really matter. So let T represent the tall allele. The small allele, we always pick the same letter, but lowercase, because the dominant one gets the capital letter, and that helps us then remember which is which. Okay, so in this case we're talking about height. So we're talking about genes for height, two different forms. Big T represents tall allele, which is dominant. Small t represents the small allele, which is recessive. Each organism has two alleles, one from its mother, one from its father. So the possibilities, we could have two big T's. We could have a big T and a small t. Now we could also have a small t and a big T. It doesn't matter which way round they are, so they're both exactly the same. We don't need to write them both down. And we could have two small t's. Okay, the alleles that an organism has is described as its genotype. So think genetics, genes. Genotype is the alleles that the organism has. The phenotype, we said earlier, was the outward expression. So in this case, the actual characteristic. So an organism that has two tall alleles is always going to be tall. One that has two little t's, well, little t is the small allele, so it's obviously going to be small. The one in the middle here has a big T, and we said the big T for tall is always expressed if it is there. and the small one in that case is ignored. Now, in your notes on page 5, it has some other terms there. Okay, the word homozygous, heterozygous is to do with the alleles. Homo in biology, or homo, means the same. Hetero means different. So this is homozygous. This is heterozygous. And these are both the same, so this is homozygous. Okay, two big T's. The big T's are dominant, so this is called homozygous dominant. The middle one is heterozygous, so obviously that means there's one of each. The bottom one's also homozygous, but it's got two small t's, so that's homozygous recessive. So we've got quite a few new words there. Genotype, as it says in the bottom of page 5, and it's worth highlighting that and write learn beside it. That's the organism's alleles. The phenotype is their outward appearance, in this case, whether they're tall or small. You really need to know the difference between those two words, because if you get them mixed up, you're going to get the wrong answers and you're not going to get marks. Even if you understand the genetics that's going on, you need to know, if it asks you for genotype, you're writing down T's. If you're asked for phenotype, you're writing down, in this case, tall or small. So if we go back to our experiment, we had the parental generation. This one was purebred tall, and this one was purebred small. So we assume that they're homozygous. In other words, that both alleles are the same. And so this one here passed on a big T to this, and a big T to this. And the small parent passed on a little t and a little t. Now, because they've each got a big T, they're both tall. So their genotype, big T, little t, and they're both tall, that would be their phenotype. Now, I'm just going to rub out that little bottom bit because we want to do a little bit of writing down here. Now, whenever these two were crossed, well, the big T goes to half of them. Well, we work out really the possibilities. That big T and that big T gives us that. We could have that big T and that little t. Still going to give us a tall plant. We have that little t and that big T, little t, and little t and little t. That is going to be the only small plant, because all the other ones have at least one big T, and therefore those three are going to be tall and the other one's going to be small. On the top of page 6, just a reminder about how we write the capitals and the lowercase letters and what they mean. We've got definitions here, and you really do need to learn these. So, dominant allele is one that is expressed in the heterozygous condition. So in other words, if you've got big T, little t, the dominant one is the one that is expressed, which in this case is the big T. Recessive allele, recessive allele is only expressed when it's in the homozygous condition. In other words, little t, little t. So you really must learn those. I know most people have in their heads the difference between them, but it's really hard to explain which is which. You can't say one's stronger than the other, or one's bigger or more important. It doesn't work like that, and this is the only real way of getting marks in an exam. So let's go on to page 7 it is. No, page 6. We'll get there eventually. Page 6 says, complete the diagram to show the cross between a pure breeding tall plant and a pure breeding small plant. So the parents' phenotypes, well, that's whether it's tall or small. So we're going to write tall. We're going to write tall here. And then the other plant, small. We can put in there pure breeding as well. It's not essential. The parent's genotype. Well, we've been using the letter T, so tall we know is dominant, so it's going to have 2 big T's, and small must be 2 little t's. It's the only genotype that it could have. Half of the gametes will have each of these. Now in this case, because they're both big T's, all of the gametes are going to have big T's, and the same over here, except they're small t's. First generation's genotype. So again, we're going to work out our 4 possible combinations here. So that would be big T, little t, big T, little t, big T, little t, and big T, little t. I'll pause at the end, you can get this all copied down. The phenotype of each of these, well, they've all got a big T, so they're all going to be tall. So underneath there, you write all of the offspring will be tall. So if you just you might need to pause the video just to get that all copied down on page 6. So once you've got that copied down, it says draw a genetics diagram to show what happens when 2 of these F1 plants are crossed. Okay, so 2 of the F1 plants, so the F1 plants would be 2 of these being crossed. Doesn't matter which because they're all the same. So if we look at the top of page 7, it asks us for the F1 phenotype. Well, they were tall. And again, I'll do this relatively quickly and you can pause and copy it down at the end. Their genotypes, big T and little t, remember? So the gametes, half of them are going to be big T, half are little t, and the same over here. So if we go down here and here, that's big T, little t, and that's going to be tall. Big T, little t, tall again. Little t, big T, tall, little t, small. Okay, so pause the video if you need to. The phenotypic ratio, so remember this is our phenotype here. So 3 out of 4 are tall, 1 out of 4 is small. We can sometimes you're asked for the ratio, so in this case the ratio is going to be 3 tall to 1 small. Or it could be a fraction, so down at the bottom a fraction would be 3/4 tall, 1/4 small. In terms of percentage, 3/4 is 75%. And obviously then a quarter is 25% small. Okay, now it's actually easier to draw it out a different way. This from year 10. This is called a Punnett square, named after a Mr. Punnett. And our cross, we had 2 of the tall plants that had the big T and little t. So, well, usually the male gametes going across top. We don't know which are the male and which are the female, so it doesn't actually matter which way around we do it. So put one of those in there and the other one down the side, big T, little t. So here we would have a big T, big T, big T, little t, big T, little t, and 2 small t's. So that's our Punnett square. Now really what you should do, I would circle that and I would write small, and all the other ones are tall. You would always write beside it or underneath it, I would write 3 tall to 1 small. There's no point writing 3 to 1 because you need to say which one is tall, which one's small. So 3 tall to 1 small. Okay. Now, hopefully that's all coming back to you from year 10. On page 8, you have got a question with part A and part B. It's asking about people who can roll their tongues, and it's giving you the letter R to use, and it's saying the tongue rolling allele, big R, is dominant. So that's for tongue rolling. Whereas then you have to work out, you know that there's an allele, you can have non-rollers, so it has to be a small r is for non-rolling. And obviously it's the recessive one, the opposite to the dominant. So use that information. I'm going to stop the video now. You're going to write out the 2 crosses. Now you need to write it out the way we've done it on the previous page. You need to write out the parent's phenotype, the parent's genotype, the gametes, and then do the cross in the Punnett square. You don't need to do it the one with all the lines, just do it in the square. And then for part B, you have another cross. It's talking about 2 heterozygous rollers. So they can both roll their tongue, but they're heterozygous. So you have to work out what that is. Do the cross, do the Punnett square, and work out the ratio again. Follow the instructions. Okay, see how you get on. So you try that. Do it in pencil, because if you make any mistakes then it's quite easy to rub them out and do your corrections.